Next topic is ascending and descending in monosaccharide. So let us first consider ascending or lengthening of carbon chain or monosaccharide. This reaction is also called Kiliani Fischer synthesis, and it involves conversion of aldopentose into aldohexose. This Kiliani Fischer method is developed in 1890, and this method involves the conversion of the aldose into a higher aldose which containing one more carbon atoms. The lengthening of carbon chain in aldose is carried out by these four steps. The step number 1 involves reaction of aldose with hydrogen cyanide. It will give a mixture of two diastereomer of the cyanohydrin, which will having a new stereocenter in the compound. Then second step involves the hydrolysis of the cyanohydrin mixture. It will give a mixture of two corresponding acids which are separated from each other by different methods. Then third step involves action of the heat on each these acid to form the corresponding lactones, and finally the reduction of the lactone is carried out with sodium amalgam to get the higher aldose, which will containing one carbon atom more than the starting aldose. So let us consider the example of the Kiliani Fischer synthesis, which will involve conversion of aldopentose into a mixture of two aldohexose, that is D-glucose and D-mannose. So here we can started with aldopentose. It will having 5 carbon atom, and it is subjected to reaction with hydrogen cyanide, and it will give two isomeric compound of cyanohydrin, which will having different configuration at carbon number 2. In carbon number 2, the hydroxy group is on right hand side in first isomer, and in second isomer the hydroxy group that is on the left hand side. So these are the two diastereomer of cyanohydrin. Then the next step will involve hydrolysis of these cyanohydrin mixture, and it will give two isomeric acids. The hydrolysis that will take place on this cyano group, and it will get converted into carboxylic group. So these are the two acids, and it will again having different configuration at carbon number 2. In first case, hydroxy group that is on the right hand side, and in second case the hydroxy group that is on the left hand side. Then in next step, these acids are heated and they will get converted into corresponding gamma lactones by losing one molecule of water. So in first case, the water molecule is lost from the carbon number 1 and the carbon number 4. Similarly, in second case, the water molecule that is lost, carbon number 1 and carbon number 4. So these lactones are get formed, and these lactones are again having different configuration at carbon number 2. In this case, the carbon number 2 having hydroxy group on the right hand side, and in second case the hydroxy group that is on the left hand side. Then in the last step, these lactone are subjected to reduction by sodium amalgam, so that finally 2 diastereomer of higher aldose are obtained, which will having 1 carbon atom more than the starting aldose. So in this case, first compound, that is the D-glucose, having 6 carbon atom. Similarly, the second isomer, that is D-mannose, it will also have 6 carbon atom. In these 2 diastereomer, the configuration of carbon number 2 is different. In D-glucose, the hydroxy group that is on the right hand side, and in case of D-mannose, the hydroxy group that is on the left hand side. So it is the ascending or lengthening of the carbon chain, in which aldopentose is get converted into D-glucose and D-mannose. So it is the Kiliani Fischer synthesis. Next topic is descending or shortening of carbon chain. An aldose may be converted into lower aldose containing 1 carbon atom less as comparison to the starting aldose. The carbon chain may be shortened by 2 methods. The first method is called Ruff degradation. So let us consider this method. In this method, the oxidation of starting aldose is carried out with the bromine water to the corresponding aldonic acids. Then this aldonic acid is converted into calcium salt, which involve treatment of this acid with calcium carbonate, and finally the calcium salt is treated with Fenton reagent, so that the higher aldose is get converted into lower aldose. So let us consider the method of conversion of D-glucose to D-arabinose. In this case, D-glucose is undergo oxidation with bromine water, and oxidation will take place at the center. Aldehyde, it will get converted into carboxylic acid part. Then this gluconic acid, it is treated with calcium carbonate, so that calcium salt of this gluconic acid will get formed. The valency of the calcium, that is 2, so it will having 2 glucose molecule, so it is divided by 2. So this calcium gluconate will get formed, and finally this calcium salt, it is treated with Fenton reagent, and it can undergo decarboxylation by the removal of carbon dioxide, and finally it will get converted into D-arabinose, which will having 1 carbon atom as comparison to the D-glucose. So it is the rough degradations Then second method is the Wolf degradation. This method involves following three steps. The step number 1 involves conversion of the aldose into its oxime by the treatment of aldose with hydroxyamines. Then second step. In second step, oxime is then treated with acetic anhydride. As a result, the oxime gets dehydrated into nitriles, and at the same time various hydroxyl groups are acylated. Then in last, the acylated cyano compound is treated with tolerant agent to get the aldose containing one carbon atom less than the starting aldose. So let us consider the example of the Wolf degradation. It will involve conversion of D-glucose to D-arabinose. So in first step, this aldohexose, it is treated with hydroxyl amine. So the reaction will take place at this aldehyde group part. So it will get converted into oxime. Then this oxime is heated with acetic anhydride, and due to which the oxime get dehydrated into nitriles. This portion will get converted into nitriles, and same time all the hydroxy group except this one, they will get converted into acylated compound. So finally this acylated cyano compound will get formed. In last, this acylated cyano compound is treated with tolerant agent so that this cyano group, it will get converted into aldehyde group, and finally these various acylated groups, they will get converted into the hydroxy group. So the final product D-arabinose will get formed, and it will contains one carbon atom less than the starting aldose. So again it will having 5 carbon atom. Next topic is determination of configuration of monosaccharide. So let us start with it. As we already studied earlier that with exception of simplest ketone such as dihydroxyacetone, all other the monosaccharides are optically active and have one or more stereocenter in their molecules. So depending upon the number of stereocenters present in the molecule, they exist in different stereoisomeric form having different configuration. The determination of absolute configuration of various stereoisomer was a very challenging task. So let us start with a brief review of some earlier development in this field. So the first attempt which is carried out for the determination of the absolute configuration of the compound is done by the Emil Fischer in 1885. He chose a standard reference compound and fixed its configuration arbitrarily, the compound chosen as a standard was glyceraldehyde. So glyceraldehyde is the compound which is chosen by him as a reference compound. These enantiomers of glyceraldehyde having specific rotation. The first one that will having specific rotation plus 13.5, and it was arbitrarily assigned the configuration and donated by D-glyceraldehyde, which will involve the hydroxy group on the right hand side. So it is the first enantiomer of the glyceraldehyde. Similarly, the other glyceraldehyde having specific rotation minus 13.5 was assigned the configuration denoted by L-glyceraldehyde, and in this case the hydroxy group that is attached to stereocenter on the left hand side. So these are two different enantiomer of glyceraldehyde. So in first case the hydroxy group that is attached on the right hand side, it is donated with D sign, and in second configuration the hydroxy group that is attached on the left hand side, so it is donated with the L signs. These are the Fischer projection formula of the glyceraldehyde. This one is the D-glyceraldehyde. It will having hydroxy group on the right hand side, and the second one is the L-glyceraldehyde. It will having hydroxy group on the left hand side. Then later on many other compounds were then assigned relative configuration by relating their configuration to D-glyceraldehyde and L-glyceraldehyde. Then later on in 1951 the Bijovet work, he can used X-ray crystallography, clearly established that the arbitrary assigned configuration of glyceraldehyde by this Emil Fischer represented the correct absolute configuration, which means if the configuration of glyceraldehyde were correct, then the derived relative configuration of other compound must also be their correct absolute configurations. Then D-glyceraldehyde and L-glyceraldehyde serve as configurational reference point for all monosaccharide. In glycerides, the stereo center is the carbon, which is last but one or penultimate carbon. So if we are having this stereomer, then this carbon atom is considered and related with the glyceraldehyde. So it is the penultimate carbon or last one carbon. Similarly, in another isomer, this carbon atom is related with the glyceraldehyde, and these carbon atoms are not taken for the consideration of the configurations. So let us consider the configuration of various compounds So this one is assigned as D-glucose because in this case the penultimate carbon atom, that is the carbon number 5, and it is the last one carbon atom having the hydroxy group on the right hand side. Similarly, in second case, the penultimate or last one carbon, that will having hydroxy group on the left hand side, so it will assign L-glucose. Then similarly in this case, this one is the penultimate carbon having hydroxy group on the left hand side, so it is assigned L-ribose. Similarly in this case, this penultimate carbon, it will having hydroxy group on the right hand side, so it will assigned D-fructose. So in this way we can assign the configuration of various compound as D and L configured compounds. Here one main point which we have to remember, that there is no direct relationship between absolute configuration and the direction of the rotation of the light. Means this D and L configuration having no relation with the rotation of the light. So let us consider the configuration of D series of aldoses. In this case, we can consider the conversion of the lower aldose into a higher aldose, in which a new chiral center is introduced next to the aldehyde group. As a result, two stereomeric higher aldoses are formed, which differ in configuration only at carbon number 2, but have similar configuration at the rest of the chiral carbon atoms. So let us consider it. Here we can started with D-glyceraldehyde. It will having 3 carbon atom, and configuration on the carbon number 2, that will having the hydroxy group that is on the right hand side, so it is the D-glyceraldehyde. Now we can introduce one chiral atom at carbon number 2. So in this case, this carbon atom is introduced in first case hydroxy group that is on the right hand side, and in second case the hydroxy group that is on the left hand sides. So now it will having 4 carbon atom. So this one is the D-rithose and this one is the D-thriose. So in both these cases the configuration of rest of the part is same, and in this case the hydroxy group on the penultimate carbon that is on the right hand side. So these are the D configuration. Now in the next step, one more carbon that is introduced in D-rithose and D-thriose. So let us first consider the case of the D-rithose. If carbon is introduced in first case, the hydroxy group that is on the right hand side, so it is the D-ribose, and in the second case the hydroxy group that is on the left hand side, so it is the D-arabinose. If we can see the penultimate carbon, the hydroxy group that is on the right hand side, hydroxy group that is on the right hand side, so it is the D configuration. So these two compound having 5 carbon atom. Similarly, if a new chiral center is introduced in D-threose at carbon number 2. So in first case, the hydroxy group that is on the right hand side, so it is called D-xylose, and in second case the hydroxy group that is on the left hand side, so it is called D-lexose. So in both these two cases, the penultimate carbon that will having in first case hydroxy group on the right hand side, so it is D configuration, and in second case the hydroxy group is again on the right hand side, so it is the D configuration. Now again the new carbon or chiral center that is introduced in D-ribose, D-rabinose, D-xylose and D-lexose. So let us consider it. If D-ribose is introduced, the new chiral center at carbon number 2, in first case the hydroxy group that is lying on the right hand side, so it is D-allose, and in second case the hydroxy group that is lying on the left hand side, so it is D-aldose. In both these two cases, the carbon number 5 that will having hydroxy group on the right hand side, so it will having D configuration. Then similarly D-rabinose, in this case the carbon is introduced. So in first case hydroxy group that is on the right hand side, in second case hydroxy group that is on the left hand side, so it is D-glucose, and second one that is the D-mannose. These two compound having hydroxy group on the right hand side on the penultimate carbon, so these are assigned as D configuration. Then similarly D-xylose are further subjected and the new chiral center is introduced in the carbon number 2, and the first case having hydroxy group on the right hand side and second will having the hydroxy group on the left hand side. So first configuration is called D-gulose and second is called D-aldose. So these two compound having hydroxy group on the right hand side in the penultimate carbon atom, so they are assigned D configuration. In last case D-lysose is subjected and so converted to the higher aldose and a chiral center is introduced on the carbon number 2. In first case the hydroxy group that is lying on the right hand side, which is called D-galactose, and in second case the hydroxy group that is lying on the left hand side, which is called D-talose. In both these cases the penultimate carbon having hydroxy group on the right hand side, so they are assigned as the D configurations. So it is the conversion of lower aldose into higher aldose, and in these cases a new chiral center is introduced on the carbon number 2, which is next to the aldehyde group. Similarly, in this way we can also configure L-aldose starting with the L-glyceraldehyde, and similar compound can be obtained in that case. Then next is the configuration of D series of ketones. The configuration of D series of ketones can be derived similarly by following same guideline as we can discuss in the case of aldose. So let us consider the examples of conversion of this D series of the ketones. So in this case the D-erythrulose is taken. It will having the hydroxy group on the right hand side on the carbon number 3, so it is assigned as a D configurations. So in this case the new chiral center is introduced next to the keto group. So in first case, the hydroxy group that is on the right hand side, and it is called D-ribulose, and in second case, the hydroxy group that is on the left hand side, and it is called D-gylulose. In both these two cases, the carbon number 4, which is the penultimate carbon having hydroxy group on the right hand side, so they are assigned D configurations. Now again, one more chiral center is introduced next to the carbonyl group. In first case, hydroxy group that is lying on the right hand side, which is called D-pesicose, and the second one, the hydroxy group that is lying on the left hand side, is called D-fructose. And in both these cases, the carbon number 5, which is the penultimate carbon having hydroxy group on the right hand side, so they are assigned D configuration. Similarly, in case of D-gylulose, the new chiral center that is introduced next to the carbonyl. In first case, hydroxy group that is on the right hand side, it is called D-sorbose. In second case, the hydroxy group that is on the left hand side, it is called D-tagatose. And in both these cases, the carbon number 5, which is the penultimate carbon having hydroxy group on the right hand side, so they are assigned as the D configuration. And similarly, we can also derive the configuration of the L-ketose by starting with L-erythrulose. So it is all about the configuration of the D series of the ketose.